which is here again, 6:45 or maybe a little after. Dr. Andrew Leslie, Assistant Professor, Department of Ecology and Evolutionary Biology at Brown in Providence. The evolution of conifer cones across time and space. So join us for that. And tonight's speaker, I didn't do my job at getting too much of your background, Bob. But tonight's speaker came all the way from Wisconsin to be with us. State botanist Robert Wernerehl is somewhat new to the New England community, botanical community. A lot of similar flora overlap, which he may mention in his talk. But as soon as Bob came on as state botanist, started showing up to meetings and just realized what a kindred spirit is. He's local to me in Worcester County, and we hang out now and again. But he's got a history of academic research, quite a bit in Wisconsin, and he's brought his passion and interest in lifelong learning to us in New England. I'm very thankful that we have him as our botanist. Sorry I didn't follow up too much on your academic stuff. Feel free to add whatever. But tonight, Bob's going to be talking about rare vascular plants in Massachusetts, natural heritage maps, and the mystery.
Thanks very much. It's really great to be here. See, Pat is back, and I remember the very first thing that I came to in the winter of early February of 2015, which was the winter of the big snow, was Pat's talk on natural communities right here in this room, and it was really a great introduction to the state. So, but thank you all for coming out. I hope I can do justice to the many topics that could be talked about here. It's a little bit about methodology tonight, so I hope that part isn't too boring. I'm not going to take a fun and fancy tour of all the rare plants and cool places. There's a little more to it than that, because this is a really good opportunity to talk about the methodology that we use in Heritage, so that you can understand it and interact with us in a certain way. And then I'll get on to the threats that we're facing here, and some other esoteric topics if time allows here. But I wanted to start with dedicating this talk to the memory of Elizabeth. This is our first chance here, and sort of our home in Cambridge since her memorial service in December. We had our one meeting in Westboro in January, but this is a good time to think about her and her passing, because of course she did so very much for this organization. And I had the good fortune to get to know her in my short time here on a number of occasions, and like everybody else, I think was very impressed with her all around. The second talk I went to, in fact, after the one with Dr. Swain, was in Cambridge, but down the street somewhere, where Elizabeth gave her talk on the state of the plants of New England. It was a big event, and I was impressed by that. So I got to go out with her to the Wendell State Forest, where she was leading a field trip, and here my photo of her describing the phylotaxy of ferns with enthusiasm and gusto, and making an impression on everyone there. It was a great hike in the Hidden Valley Memorial's forest, which is there on Arthur Cronquist's old haunts in Wendell. So a really neat hike. And Ann and I were the very last ones out of the parking lot that day, and the second to last out of the parking lot was Elizabeth, which meant that we got to see her make a last impression. This was the last time I saw her alive, and it was the bumper sticker on her car that gave us the last impression of her constant cleverness and intelligence. And I would like to think that, yes, Elizabeth, you now have many vehicles and many songs that we all are carrying on, because it takes a village to replace what this one single woman was able to do single-handedly.
Bob, thank you for mentioning her. I actually brought 30 of the programs from the memorial, and I'll pass the envelope around if you'd like to take one. There's 30 anyway. I'll bring them in now.
So my topics tonight, just to start with, to go over some of our methodology in heritage and ask some questions about it that I think can improve how we conceptualize what natural heritage does and how it fits in to the overall botanical community. And then the second topic will be trends in rare plant populations, or perhaps in some cases lack of trends. And if there's time, and I should watch the time because this talk isn't a short one, we'll get to a theoretical question of how many vascular plant species should be rare in Massachusetts. It's an interesting scientific topic. So I'm just going to, because I knew Brett wasn't going to cover it in depth, and because I am really relatively new here, I just thought I'd touch very briefly on my work in the Midwest, not just Wisconsin, but this is just a small part of what I did there. I led the conditions assessment team for 2 years in the summer, and got to some, what was probably to you very familiar habitats. We have good bogs up there, extremely similar flora in the bogs, but of course the wetland assessment takes on all sites. So we had these two, cornfield edges and cattail, pretty yucky, and run into some cool critters, Cecropia, Fitstar, Instar, Onspirea. And we also ran into some of the critters that you might expect that we would have run into. Curious about what the wetland scientists are doing in their pasture. They were very friendly. And then I led teams with, I had a consulting firm for a while, and for 11 years we did surveys in the summer in these 3 national forests, the Chippewa and the Superior and the Ottawa here. So in each of those are about 1 million acres on average. So pretty good chunks of land, fun places to explore. You're out day after day, walking through the woods, and occasionally see some plants that may be familiar. This is Nufar, you call it microphylla. It's still called Nufar pumila in Michigan. And we made a find there. It hadn't been found in almost 100 years when we found it. And then the mysterious and usually well-hidden goblin fern, Batricium mormo down here. This is in the Chippewa National Forest. It's sort of the hub of the population. And there's one in my hands there, the goblin fern. Then I went on to do some PhD research on remnant prairies in Wisconsin, in part. This is one on the Mississippi River, a dry prairie. We have a lot of good remnants left in Wisconsin that have been kept intact more or less for the past 5,000 years or so. And so they have a really good and well-studied flora. These are some of the other sites. Some tall grass prairies. I know Nancy did her research in Arkansas. Tall grass prairies. There's a dry, wet mesic prairie with Lytris pycnostakia. And I also did a lot of greenhouse work with the Lycor photosynthetic measurements. We did treatments of 4 species of plants, trying to flood them until they kick the bucket and trying to dry them out until they kick the bucket, which is not easy to do with prairie plants. And meanwhile, testing them for instantaneous photosynthetic rates. And we have the same species here. The one that's not labeled is Schizocurium. So, and Bootaloo is here, except very rare. It's Bootaloo curtipendula. So that's a drought and a flood. But I don't want to take too much time on this. So let's see, these are my mesocosms that my friend had. He was a honey wholesaler, and he was recycling, having all these barrels crushed. And then I said, you know what, I could use those. So we filled them with a mix of soil, planted 5 species around in a star pattern on these, and grew them out for a year under differential conditions. And they were supposed to be growing for several years, but apparently I was praying to the wrong weather god. I was praying to, a little unknown to me, to an old King Louis. And he said, après-mal le deluge, and it rained the next year 14 inches in 8 days and flooded everything out. So it became one big flood experiment, which was kind of interesting. The Spartina had a great reaction to that. Everything else kind of called it quits. So that's a little bit about what I was doing before I came here. So I'd like to move on to some of the things we do at Natural Heritage. Some of you may be somewhat familiar with this, but there may be some details that you haven't seen here. And as I said, I'm sort of asking you to reconceptualize our approach. And as I was pulling this talk together, I myself sort of became to reconceptualize what it is that we do. We want to be sure, we need to be sure, because our species, if they're state listed, when they're accepted into our records, fall under regulations. So that means that those people who would like to build a dock and our rare plants are in the way, they have to come in for environmental review, or it could be a bike trail or a horse trail or anything else like that. And so we need to make sure our records are accurate. But we're doing this generally without collections. So we, in a sense, become a type of virtual herbarium. So you're going to see how we go about doing that. And you can assess for yourself whether we're taking the job seriously enough. These are some of the screens that we see, that Carol and I see right now, before us, Brian or Melissa, perhaps, when we're accepting a record. And I call this quality control step 1. These are the questions that we ask you, the observer, and now we're, as the records come in, checking to make sure that you, the observer, who's filled these out, has answered these questions. And all of these things help us determine the accuracy and confidence of the record. What reference you used, and I was actually approving this record as I went along. This is a watch list species, so I didn't have to redact it. Houghtonia, you'll see a picture of it in a bit, submitted by Tim Simmons. So I was bringing this record in as it came along. Anytime we want, we can click on this little button and bring up the map right away to see whether the habitat is accurate or not, and so on. And sometimes you're in the field, somebody else made the identification. You may not have been as certain of it, but somebody else who's even more familiar, you should put that down. And it helps us to be certain ourselves. And then these are the wonderful data that you provide us that are so very helpful in helping us assess rare plant populations. Nothing particular about accuracy there, but it just really helps us out. This is about accuracy and about virtual herbaria, as I'd like to think of ourselves. As some of you know, seen me talk before, I do tend to emphasize the importance of these type of photographs where a ruler is involved. And it's fortunate that your president here, Brett, has a fine example of submitting records with excellent use of rulers. And you'll see later on in some of my own photographs, the rulers. Now, you might think, this might not be necessary for these, and in some cases not. You can certainly submit others without rulers, right? In addition to the ones with. But let's say a species is reconsidered in the taxonomy. What if a subspecies is split off? Or what if new taxonomic information comes in and reconfigures the dichotomous key or the actual alignment of the species? Since these records and these photographs are permanently attached here, we can go back, and if we needed to, we could, with accurate photographs, reconfigure the record to whatever new paradigm it might be. So all of those things are important. In the case of Houghtonia, you don't really need a ruler. It's one of my favorite species. I like to think of it as being drawn by Dr. Seuss. He was playful. It was a great year for it because it's an unusual winter annual, so the seeds have to come down. We had an extremely dry year in 2016, so the seeds came down in the mud and germinated. Then they were covered by the good supply of water in 2017. And so we got a number of new records of Houghtonia, including from Tim Simmons here. So, and this is the habitat shot, so we can kind of evaluate it from that, but also we'll bring up the aerial to check that. So now we move on to quality control step 2, so to speak. And this is our internal checklist. So these are things that you almost certainly would not have seen unless you worked in Heritage before I got there. So there's 6 steps involved, and these are the questions that we have to look through as we're approving the record. Does the person have documented experience with this particular taxonomic group? In the case of tough choices, we might want to go to our other records and see if that person has other records for that, or seeing if they've just been in the field with people who made those identifications. And if not, it's not that we would reject it, but we would have further concerns or questions about it. We want to make sure that it's the appropriate habitat and it's within the known range, and that the phenology is right for this. And then we could hit the accept button, and it generates, with the help of our fingers, a beautiful email to you thanking you for your fine work. And we really do mean that when we send you those emails, because all these records are very helpful and very important. We can also have a couple other choices that I don't have down here, but we can just keep them on hold and give us a little time to ask more questions about it. Like, if you didn't put down who you were with in the field, or some of the other conditions you might have had, or do you have any other photographs, or did you take any measurements that you didn't put down? That's pretty rare, but we are able to sometimes find out more on something like that that would allow us to later on accept that record, which can really be great. Quality control step 3 now passes to our data staff after we accept the record. It's nowhere near done. It has to be checked by not just one, but 2 data staff independently and separately. And they're going to set up the element occurrence feature. As they do, they actually create these features on the map, and this goes right into our biotics database directly. There are 3 choices here, but I just give them 2, the point and the polygon, and they deal with locational uncertainty, a term that they like to use, which has to do with if you're using a handheld GPS device in the field, if you did not make a note of how accurate it was at that time, then they have certain rules that they follow to map these things out so that we don't send people to the wrong place if they need to relocate them. And then once the first person gets through, they set these 2 tags to not reviewed or incomplete, and then it goes into a digital bin, so to speak, and the next person has to come along and pull those out and then look through every single step, make sure it was all accurate, and set these to the QC status of data, QC status of map to pass before they're clicked through and they're accurate. So and then there's one final step here that it creates this unique record. So this is really similar to what herbaria do with their records. They create a unique digital code that stays with that record forever, so it can be looked for later on. And you see a lot of familiar names here. Carol's down here once, Tom Rowinski, Chris Bueller, Robert Burton. We don't know what that is, but there's no doubt some interesting plant. That it also would allow us to, at least in some cases, look up, say, whatever records you submitted in a given series of years if you needed that information. Now, we get a lot of records because people are fairly happy, with some exceptions, like this guy over here, with BPRS. Now, these data come from all taxa. So people have responded who submit animal records, invertebrate records, what have you, bird records, as well as plants. But the vast majority are happy with our new electronic system. And then there's some 14% are neutral about it, and only a few are kind of down in the dumps about it. But as a result, we are getting more. Now, this is all plant records only. This is no animal species. So you can see is the trend line there, more every year. And in 2016, we had quite a big year with over 600 records come in. The orange are paper forms. So you can see we have a lot of work to do to keep up with that. And but that's a good thing because all these records bring in new and interesting information that I'll try to display later on in the talk, at least a few aspects and tangents of this sort of information here. Now, as I was saying, this is a reconceptualization experiment. You're now thinking of us somewhat, at least, as a concise and accurate and long-lasting digital herbarium. And one thing that, there are many disadvantages, of course, to the idea of a virtual herbarium. You can't check the actual plant. You can't see whether it's hirsute or whether it's scabrous or whether it's this or that. All those features that are very important in actual specimens. And we do collect and store some actual specimens ourselves. So it's not like we're at all against that. We're strongly in favor of it. But we have to do our work mostly digitally, mainly because we work with rare plants that way. There are some advantages. Unfortunately, I can't show this because the computer won't swap from one bit of software to another. But if you've heard of photospheres, you should definitely try them out. They're you do them with your phone, with certain Google phones, or I'm not sure if it's on other platforms in Android, but you can hit this photosphere button, and then it tells you when to stop and when to start. And so I can stand here in the spot and looking at my screen and a little blue dot, and it goes clink, and then I go here and go clink, and then I can go all the way around. And then I can go all the way up, and I can go all the way down. And then you press end, and it stitches the whole thing together, and it's a complete 360 in both directions photograph. And it's really, if you want to see this afterwards, by the way, please ask me and I can show you. It's really cool because then this is just one image from there. But you can grab the mouse and you can start pulling it around once it loads. It has to load off the internet in your browser. And so it's this weird hybrid of a still shot and a movie. So you pull it around and all of a sudden you're seeing there, and you can pull it around with your mouse and you're seeing that. So you get the complete view of the habitat from that spot. And then, as you might want to, in many cases in Massachusetts, assess the canopy, you can look all the way up and then you can say, well, it looks like about 70%. It's mixed hemlock, it's mixed beech, it's mixed oak up in the canopy. And there's areas for the sun to come in here. And then, of course, you can go back in 5 years or 10 years and pull that same photograph up and compare it to when you were, whoever was last there recording that. So it's a really neat method, and I know I store them, and I hope to figure out a way in our database system to incorporate those in. So we've talked about a couple of aspects of the process now, and now we're going to talk just a little bit about heritage, natural heritage system approach nationally, and what's been going on for 30 or so years, and whether it's good or whether it has any difficulties associated with it. And of course, you can expect that it would. This is a plant population that is considered one element occurrence, and I'm going to talk about that as we go along here. They're not rigidly and hardly defined. They're fairly well defined. So an element occurrence consists, of course, of an element, and that is because Heritage came up with this long ago. That came out of the Nature Conservancy. They had a very practical ideas in mind. They weren't interested in, how can we academically assess all these things. They wanted to affect conservation on the ground. So they took it as a biological unit of conservation, attention, and action. Interesting definition, and it led to these advantages of being able to, for example, use natural communities as elements and then element occurrences. So or vernal pools or hibernacula. So you could ask yourself, how many poor fens or how many hickory hop hornbeam communities there are in Massachusetts, because it became an element occurrence. And you can, if you go out as Pat did and study them, you can tally how many occurred, so we can know how rare they are or what sort of features associated with them. And that's a useful aspect. I'm going to get to some of the disadvantages in a bit. An element occurrence is the fully occupied space that contributes or potentially contributes to the persistence of a species at that location. it's always good to sort of remember that definition as you're trying to map out an initial element occurrence that may be a composite of several sources. And so if you know what one is, you also need to know what one isn't. So when you're in the field, you know when you come to the edge of one, and you can say, well, this one ends here, and there's now a gap before the next one would occur. So here it is mapped by data staff, and these are element occurrence representations. They're sort of blob-like things that come from those locational uncertainties that as they got mapped. This one is unusual in that it's 3 kilometers all the way across there, and we're going to go into just a little bit more depth on this one because it brings up the advantages and disadvantages of this system. So these are these initial maps. There's about 30 of those blobs there, but underneath that, yeah, I know you all know what this is, but don't say it because it's on film and we protect it. You're not allowed to say it. Yeah, so underneath those blobs are these sources. These are source features. Now we're looking at 90 source features. And they are mapped and the data is gathered in from those in an unusual way, and I'm going to get to that in a minute. But while we're on this screen, what we need to ponder is what you really need to know when you're thinking about rare plant numbers in Massachusetts. Because this element, this single element occurrence could have been and may still be defined in different ways. For example, 5 years from now, we may decide that there's too big of a barrier across here, and we may decide that the soil type on this side is different, that there's a south aspect is too strong here, and there's a particular wind that comes up here. So essentially there's a barrier across here. So we may circle that up. Then we may decide this is nice and flat here, it's got special, like maybe there's some pitch pine remnants or something else. So we're going to circle that up and call that a single one. These are on a really steep slope, all clustered together. So we're going to circle that up, and we're going to say that there's physiognomic barriers between those. Then we're going to go in the north and we're going to say those are really like north aspect, they're a lot cooler, they don't really have that much to do with the ones to the south. So we're going to make 4 element occurrences out of those. That could be 5 years from now, 10 years from now, we don't know. But all of a sudden one day that means you as the interested party and thinking about rare plants in Massachusetts, one year you look at a record and you say there's there's in the whole state, there's say 5 element occurrences. There's 5 locations of this plant. And the next year you look, now all of a sudden there's 8. How did that happen? Nothing's changed except the definition, and that's allowable under the definitions of element occurrence. But what's even more likely is it started off when Paul Summers was the botanist, right, Paul? You just had these down here, then somebody went and found these up here. So they were way too far apart. They were 2 element occurrences. Then somebody came in and found all these, and then finally they found these and these down here. So then we saw that it was in fact a contiguous population. So 4 disparate element occurrences then get mapped into 1. And then you, the interested plant party, is looking at your notes on Massachusetts plants, and all of a sudden your favorite plant went from 5 EOs down to 1. You're thinking, oh man, this poor plant is taking a beating, but nothing's changed. So that's a really and something I really wanted to bring up, that we have to look in some cases deeper than the numbers, and we may have to think about how we want to report out these numbers, and we certainly may want to change how we're storing these numbers, because and here's what I was talking about here. In the past it's happened, and so you recognize this little type of screen is what data staff see, and this is a note from one data staff to another. In 2001, EOs 13 and 28 were merged in this record, and some information about the source. Only the Northeast Cove was surveyed, and so some information there as to the fact that this actually does and did happen, and still does happen. It's part of the natural process here. By the way, did I see art here? Yeah. This is the Pyrola acerifolia. We found that field trip last summer. I just wanted to brighten up that otherwise dull slide.
So it's occurrence 13 and 28.
Oh, yeah. Yeah. Okay. So how do we store this particular data with all these sources? We store it in a bad place. And I'm really going to hopefully be able to change this. We store it in a text field, EO data field. It's taken me like years to figure out why it was so sort of difficult to analyze and conceptualize these things. It's because this is how it always got stored. And here's what that looks like for that record with all those 97 sources. And it's not even the end of that. That's only partial, right? Look at this detail. Sources 70 to 73, approximately 250 were there in leaf over 3 years. Then we go down to source 14, 38. Then we go over here, source 67. And you're trying to read through this and think, what the heck is going on? Source, you even get more defined than that. Source 21B, not 21A or 21C. So it gets really complicated. And it turns out that there's another way to do it. And I just found out about this last week. I mean, nobody ever told me about this, but there's a source field with a visit component. So they can be stored in separate chunks. And they would then come out, what I've always lusted after is to be able to make rows on a spreadsheet with all this information. I don't want to sit down and have to pick these line by line, date by date, and create my own spreadsheet from it so I can analyze what's going on with the population. And that turns out that's possible. They've been doing it for a couple other animal records, and now we're going to hopefully experiment with doing that for plant records so we can, that answers the question, how can our system be improved? And it's going to be interesting to go on. There's other major improvements that we have going on with the way we collect data using equipment in the field too. So it's still an exciting time to be a heritage botanist. So we've covered all aspects of topic 1, and we're going to jump to topic 2. And we will take some tours now of things around the state. Trends in rare plant population. I'll tell you when to cue the dark foreboding music. It's soon, but not yet. Let's start off on a happy note. And then just a little line that if we get to the third topic, that's what we'll be about. Happy note. We picked up on this plan from 1996. One of the few times the federal government decided to spend money on rare plants. Woohoo! And not just blow it all on those furry animals and feathered animals and all those other creatures out there. And so actually Alan Weekly wrote this plan in 1996. Some of you know Alan. Tremendous, incredible botanist down in North Carolina. And so there we are standing on the beach in 2016 on Nantucket, pondering what our sites are. Most of these are Fish and Wildlife staff, except this is Michael Coons from North Carolina Botanical Garden, and he was the one that grew out some of these sea beach amaranth, which is an annual. So seeds were collected from up and down the coast. We got the northernmost batch that was kept separate. It was from northern New Jersey, and they grew those out down there so they could produce enough seed over a couple seasons and have them ready for this year, which was 2017, and here's where we decided to plant. So this is one of the things that I've been sort of favoring lately, and some of you that are here tonight are involved in other restorations, Paul and I read out in restoring some Amelanchier, and we've been doing a little bit with Liatris, and here there are arguments against doing that, but I'm more in favor of doing that for keeping the plants alive as long as we can here while we face the next couple decades, which are bound to be quite tumultuous in terms of weather and change here. So these are 2 spots on Nantucket where we planted them with help from Nantucket Conservation Foundation, and a little bit on the Wildlife Refuge, but also property owned by TTOR, the Trustees of Reservations. And then this is us June 22nd on Monomoy, full day of summer with full sun out on Monomoy Island. And there's Michael Coons again, and this is Dale Suter, who's out of, I think, South Carolina Fish and Wildlife, who's been working on this since 1996. Amazing guy. And so there we are. And then now we're moving out to the plots, which went for like a mile down the beach. It was a pretty amazing day. We had little peat pots, and we got them wet with this water, and put one seed in each one, and put 150 in each plot, all spread out, and we had 15 plots. So we did a lot of plants that day. And then Michael came around with his broom and just covered up the, the plants. And some of them survived Hurricane Jose, again, praying to the wrong weather god, apparently. And Hurricane Jose came along in the end of September, you may remember, and there was a lot of overwash. But I took these photos here in about the 12th, I think, of October, and they were doing okay. So I think some are going to survive and reproduce and start a new population, which is part of the big federal recovery plan. That will be our 4th federally listed species, so that'll be a cool thing. We're staying ahead of the other taxonomic groups. That's cool. They have fewer federally listed ones. How did I learn about this plant? Well, I took a little tour of Fire Island, which is on the south shore of Long Island. This was in 2015, and this is the habitat here, and you can cue the foreboding music now. Why are these cages here? And what is that? Well, it's Amaranthus pumilus. They're being caged on Fire Island because of voracious deer that would come along, walk along there. They apparently can't be hunted there. And they see this tiny little green plant, looks kind of good, tasty, and the staff told us that they'll just, reach down and they'll pull it right up by the roots and nibble it down, and it's gone. So not a good thing. In 2013, some TNC scientists put together this article and asked this important question that it's really important for us to consider whether this is the case. And I'm going to give some evidence in favor of their line of thinking now. It doesn't mean that climate change isn't important. It's very important, and I've been touching on that already. But Bambi here is definitely doing a number on us here, and we don't have nearly the dollars that climate change adjustment people have, climate change assessment, climate change planning, all the thousands, hundreds of thousands of dollars that goes into it, even just within our own state. And we barely have enough staff in wildlife to spend even a few months assessing deer. These are some of the reasons that they set out in the 2013 article, which is readily available online and well worth a read. It's fairly short, but to the point. No other threat to forested habitats is greater in the eastern U.S. at this point in time. Not lack of fire, not habitat conversion, not climate change, only exotic insects such as emerald ash borer, hemlock woolly adelgid, and disease have been comparable in magnitude. I have my own data that I've been gathering at all these spots around the map. And these are places that we're going to do a short tour of a few of them now, so I can try to make the point about deer, because I think it's extremely important thing to know. And some of these sites are ones that they're set up big fences in order to defend themselves from these voracious critters. Others are ones where we're busy setting up fencing to defend the plants for them. And others are just sites where I've seen heavy use and browse by deer. We're going to start in Nantucket and go to Medfield, and then, or the Blue Hills in Medfield, some sites in Gloucester, some sites with the Sudbury Reservoir near Framingham and Marlborough, a little bit on the Quabbin, and then out to the Berkshires. And these are all sites that I've been to and seen myself. I studied deer herbivory a lot in Wisconsin. It was a huge issue there. Deer populations are even much higher there, in part because of the richer soil. It's an interesting situation. I talked to Arthur Haynes about this recently, and he said in Maine, they really don't have the problem because the hunting culture is so strong up there. But we've started to lose that here. In fact, it's being lost in general across the U.S. So Nantucket, we have Navalis serpentarius in beautiful bloom, some of the best populations in the state and the best on the island, because the staff out there tell me that anything that's outside of a fence gets nibbled up by deer right away. Well, why is this one fenced? This is why. It's at the airport. They had a huge fence. They don't want the critters running across the runway. And they fortunately left enough space and allow some great native plants to grow, and some very nice sandplain grassland habitat within the airport there. Metfield rhododendrons, rhododendron maximum, state threatened plant. Carol and I were there in March of 2016. We saw heavy browse lines for quite a few acres where deer had nibbled off everything to about this height, so you could just look down, you could see like someone had come through with the shears, and in the center of their population, they've had to erect a huge fence to protect and see if they can get any regeneration at all of this thought to be native population. Here's Amanda and Carol and myself setting up a fence in the Blue Hills so we can protect this plant from being gobbled down, Ageratina aromatica, state endangered plant that we have started hunts in the Blue Hills now. It's one good thing my agency has done. I wish they would do more to bring down the deer population, but we did see blooms of this for a couple years, and now all of a sudden these plants are back and we're trying to protect them. Now we'll go up to trustees of reservations property where Magnolia virginiana is, Sweet Bay magnolia, and in Gloucester. And you can see the one small fence around one plant, but what you can't see, and I didn't get a good photograph, is an 8-foot tall fence around about 5 acres of this swamp to try to protect the magnolia, which the deer have just eaten up there for decades. And it's really starting to come back thanks to their efforts, and they put a lot of time and effort in, partly thanks to Russ Hopping there. Now we'll take a little tour of the Sudbury. This is one chance I got out to go with 2 of our staff in wildlife. We sort of train each other on deer herbivory, but I'm usually identifying the plants that the deer are eating. And you can see where this, close by here, it's just, what, 40 minutes west, north of Framingham. There's 495 there, there's Marlborough. And it was a pretty amazing day in a bad way. So here's one of the bonsai trees that they've nibbled off year after year. So it just gets to stubs like that. This is how you tell rabbit browse from deer browse. Rabbits have top and bottom incisors, and they cut things very sharply at an angle. It's very clear. And so when you're lowish to the ground, you can tell whether it's been deer browse or rabbit browse. Here's chalice by, I think, a red maple with a lot of stump sprouts. We see these a lot in high deer densities where something might come up and then it's nibbled off, and then so it keeps resprouting there. They even ate white pine. It's still at Sudbury, to the height of my shoulders there, all the way up, and then they stopped after that. So that was a ravenous population. Another bonsai tree here, year after year of eating. Now, if they are eating all these woody plants, I ask you, are they eating herbaceous ones in the summer? You bet they are. Kilograms of green plants every day. They're voracious. Low bush blueberry, and lots and lots of deer browse there. Now, this is one of the effects of high deer numbers over time, because ferns are very resistant to deer browse. They have strong defense mechanisms built into the chemistry of the leaf. And so hay-scented fern becomes very dominant and very abundant, super dominant, super abundant. Nothing will come through. There's chalice again in the middle of that forest with absolutely no regeneration. Now in the Heart's Content forest in Pennsylvania, where it's been resurveyed, first by my friend Tom Rooney, after 65 years, he went back and resurveyed what had been a fairly rich forest in western Pennsylvania. When he went back, because of the deer populations there, it was mostly hay-scented fern, and in the Hemlock Beach part of that forest, there was an 80% species loss, mostly due to hay-scented fern. So watch out for that. Deer have huge effects. So we're out to the Berkshires now, and we're fencing more stuff. This is with the lightweight nylon fencing, and Tony Gola's trying to fasten it to the ground there. And what are we fencing out? Showy lady slippers. Do deer eat them? You bet they do. They're tasty. It's like a big old flag hanging out there. Come and eat me. half a mile away, yellow lady slippers. That's why we listed all 3 varieties as endangered this year, is in part because of the voracious habits of our furry friends. Now, you think we know already from the amaranthus that they'll eat short stuff here. You think they wouldn't eat Goodyear tessellata now, would they? Yes, they would. There it is. See how ragged that is? That was in the Quabbin. Crazy. Liatris out in Hamilton in the northeast. Another clear, evidently deer browsed. And this population had to be fenced as well, or the deer would have gobbled it down. So what can we do? I'll finish up this little mini topic of deer. Tom Rowinski's book is helpful. It's a free download. We have some printed ones in the office. It helps train landowners and land managers how to think about these things. And of course, Tom is ever, if you know him, he's ever vigilant on this topic. It has some very good photographs in it too. Now we're doing our own data sheets in the office. So we have Dave Steinbrook, our deer biologist. I'm very grateful for him. You see, he's not just a deer guy. Let's keep the deer populations where they are. He really wants to understand what's going on, and it's really been fruitful. And it's unusual in a heritage where endangered species people would work with wildlife people. But Carol and I have both worked with Dave quite a bit, and it's very productive to do that. And we're all benefiting quite a bit. So this is some of the data sheets Dave came up with, including this part here. So we're starting to gather and accumulate data on this very important topic. What are some of the other threats? Well, here's a short list here. I'm going to cover just a couple of them here. We don't have time to cover them all, but we can cover a few, and some are interesting. And I wanted to start off with a few that I think are somewhat stable. Maybe because these first 3 I'm going to show, deer cannot really effectively get at. This is Lycopodiella up in Winchendon. It's on very deep peat. I know because I went up to my hip in a peat. Now a deer is not going to be walking around on that stuff. They've got much narrower feet than I do. So, and I think, Paul, I think you know this population too, and it just seems incredibly stable. We were just there this year, and unless somebody wanted to fill that wetland, it's probably going to be there for a while. Carex corderiza out in the town of Richmond, on the New York border, in this totally sedge mat, floating Carex lasiocarpa, floating sedge mat, very stable. Now this is not a common plant even there, but and this was something it hadn't been seen for about 18 years, I think. We went out and found it with really not too much difficulty. It's never going to be abundant in here, but there's no signs of change. There's plenty of water. The conditions are really similar. The habitat is excellent. The sedge mat, the diversity is great. Phragmites appears to be kind of a long ways away. So we're lucky to have Maddie as one of my interns this summer. She's Kathy Harris's student up at UVM, graduated this year. She had black on that day, so I thought it was useful because Carex corderiza has these long but very evenly spaced rhizomes, so it was a fun way to get her to display the, and when we were done with this, you can just lay them back in the water and they'll be just fine. So it was a really cool plant. There's only one population left, but it seems incredibly stable there. And then finally, one more example of stability is probably this Asplenium montanum on this huge rock. I'm not going to say where it is, but you probably know. And the only thing that could really get after that would be rock climbers, and I don't think they're allowed there. So that one's surviving just fine. We had a pretty good population count there this year. So those are some examples of, to contrast, what I see is some of the things that are at risk here. So fire suppression is a topic that botanists can talk a lot about. We have a lot of plants, and we have a new fire ecologist now. New, but she's been around a long time, Karen Kalju, one of our state's best botanists. Really lucky to work with her. She and Tim came up with a list of about 90 plants that they consider to be plants that need fire on the landscape. And conversely, if they don't have fire, they will be going down. in general, you would expect, and my advisor did a great paper in Science on this in 1996. Came up with expectations and found that in Wisconsin prairies, those that were burned favored those of small stature, light seeds, and end fixers. And if you want more details, you should read that paper. It's a lot of citations in Science. It's fascinating work. So here's an example, Quercus stellata. I was reading a paper on this yesterday. This plant in Illinois, there's some really old ones, and so they can saw them open and do fine-scale dendrochronology, find out the fire return interval. This thing was reproducing just fine in southern Illinois 200 years ago with a fire return interval of less than 2 years. It's amazing. And so this was a new element occurrence out on in Brewster. A really cool plant that I didn't know a whole lot about. So it doesn't come into Wisconsin. So Tim was educating me there on Quercus stellata. And it's a watch list species, so we do track it. And he just found a new one he was all excited about near one of the Pine Barrens offices there. Okay, so that's fire suppression. We touched on that. Let's go to climate warming, sea level rise, climate change issues. Yes, they are important. Okay, here's your chance. What's this plant? Very good. Yeah. Wow. Is that Louise? Man,
Yes,
you nailed it. Great. I wish I had my little chocolate to throw back there.
Did they change the name?
Oh, did they? Yes, they did. All right. Spartina is part of the Spirabolus. So now it's, yeah, they did just change that. You're right. Mm-hmm. I try to ignore those as long as I can. Amazing plant. But what's the issue here? Well, the issue is, the topography. This is at the edge of the marsh. There's the main salt marsh down there. And it's in, most of these populations are in small topographic relief valleys. So as sea level rises, they're going to get pushed. And they're only going to have about as much room as this table pretty soon to get by. So it's going to be tricky. I have seen them in a little deeper situation, but those are going to get flooded pretty heavily too. Cool plant. Carex exilis in Linfield. This was in a beat up, very urban wetland, but it's still an old record that we refound in 2016, hanging on. But this is really a northern distribution plant. These are interesting maps that you can get if you click the map button on the Consortium of Northeastern Herbaria. And so you can see it goes way up into Canada. So we're at the southern part of the range. So, we're probably going to lose that soon. And that's sad, because it's a beautiful sedge. Awesome sedge. Now we've got one plant that I don't know, but it faces the triple threat: beach driving, climate warming, and sea level rise. And this is Atriplex gabriuscula. I thought I had identified one. I had good pictures, but I sent them to Arthur, and he said, no, it's not. But it's pretty rare. We're going to see in a minute how rare it is here. But you can see, again, very northern distribution and on the beach. So it suffers from these people. Now, this is the only way I can enjoy my beach, to hop in my four-wheel drive, lower the tire pressure, and go plowing through the sand. You see all this completely lifeless. That's out at Race Point, where just like a month or two before that, it had been full of life, full of nesting piping plovers, full of tiger beetles, full of all kinds of interesting life. And as soon as they take that symbolic fencing away at the end of July, boom, out they go, tearing up the place. Nothing can survive that. No CB chamaranth, no Atriplex, no Polygonum glaucum, all kinds of good stuff. It's very sad and shocking to me, and we need to. It hasn't been all that long since people have been able to do this, but now they think it's their God-given right. Something has to change there, and I hope it will. I'd like to see more national park type status where people can get permits to hike in and camp out there. I'd rather have some people camping out in tents and hiking in than all these people. But all right, so here I get to one of Alexei Zinoviev's new map, Atriplex gabriuscula. Here's the distribution in Massachusetts. This clever guy in the back there, Alexei, has figured out how to pull records in from the CNH portal, Consortium of Marine Research Herbarium, where all the records go from here, from Harvard, and also from UMass Amherst. All the local herbaria gets pulled together there. And then, what's so great about this, if we can compare slightly to this map, you see they're all just the same, plain old ordinary red dots. What he has done is he's taken a conservative after 1980 as current, and those get marked as red. This one means they have a photo of it. And then the rest are all pre-1980. You can see this plant was, not that super rare, but is now nearly gone. We're going to look at some more of those maps later in just a little bit. Here's the one you would never guess. I get an email every spring from some of the staff in Cape Cod National Seashore because the geese are gobbling down Aurantium, golden club, out in Provincetown. And just at the west edge of town, there were some big shallow lakes there with a lot of this plant. And the Canada geese are going, well, I shouldn't say hog wild, but apparently what's good for the goose is good for the gander, and what's good for the goose is Aurantium. And so they're feasting on this stuff. And we need to figure out a way to stop it. It's definitely not a laughing matter. When we look at heritage, this is our first chance to really map the mystery, as I said we were going to do. These are all our records, all the element occurrence records. And they can go back, in some cases, we pull in records from herbaria. So some of our records are 19th century. Most of them are more recent. But this is throughout all time. So 23 records, and now these are all that are current, meaning in the last 25 years, a less conservative but regulatory length of time where we can regulate these plants. After 25 years, no regulation. So that's how I know where these are. And there's only 4, so it's very sad. One is near us in Princeton, Holden area. All right, so Canada geese herbivory. Who would have thought? But there it is, and it's probably going on a lot more than that one plant. Nitrogen deposition. This turns out to be a more interesting story than you might expect, and I'm going to show some really cool maps that you can get, all of you can get online if you love maps as much as I do. Here's the start of 6 maps. This is hydrogen ion wet deposition. Here's the website. 2015. Look at Massachusetts, not so bad, right? We're not bad. We're about the same, maybe 10 times what it is out there. This is 0.01 and we're 0.11. And but it's still very low in hydrogen ion kilograms per hectare. Now we're going to take a step back in time to 1985. And we're going to do that for 2 other elements as well. So here we go back. Yikes. Talk about the issue of legacy effects and extinction debt. Some of our plants may have essentially undergone physiological extinction back then, but they've been hanging on since now. So that, and now we're sort of paying back what we did back then. And this may not be the worst. Here's sulfide deposition. Sulfur, wet deposition as sulfate. And not so bad anymore. Really not too bad, about the same as parts of Maine. Not as good as out west, but let's take a step back. 1985, bam. Look at that. Way over 24 kilograms per hectare of SO4, which turns with rain to sulfuric acid. Think of the effect of that in the soil and the lakes. Incredible. And in soil, not just in lakes, but any of this acidification puts heavy metals into solution. And it does that in the soil as well as in lakes. So it's scary stuff. All right, one more. Nitrate ion wet deposition, 2015. We're not so bad, although 7 times worse than Nevada. We still are pretty good. 7. Nevada's, parts of Nevada, 1. And this is just nitrate, so it's just one of several components of nitrogen deposition. Ammonia is big as well. Let's step back to 1985. Bam! Look at that. More than 20 kilograms, or right around or more, 20 kilograms per hectare of NO3 landing there, and inorganic nitrogen, which is readily available to all kinds of plant life. Now you think, what does that do? Okay, I read somewhere that in England the chalk grasslands are suffering because of nitrogen deposition. Yes, they certainly are. But what does that do here? Well, there's some really interesting research going on at Harvard Forest with Sarita Fry out of UNH. And this is really interesting stuff, because nitrogen deposition is linked to manganese availability, and manganese availability has an effect on fungi. Isn't that bizarre? Here's what she found. Manganese is an important nutrient for regulating lignin decay enzymes produced by fungus. Turns out there's a great paper in 2000 in Ecology on this too, so it's not a new idea, but they knew less about it then than they do now. They're finding out more. Manganese is really interesting. It's right next to iron on the periodic table, and very rare, but apparently very important element. So, you know, long-term under nitrogen enrichment is there's a reduction in the manganese, and that can play an important role. Well, what group of plants do you think uses fungi in the soil more than any other group, besides maybe Batriciums and Ophioglossacea? Orchids, yeah, exactly. So now we'll map the magic again. This is all our 549 element occurrences of Orchidaceae. It's pretty cool where I work. I can press a single button to generate this map. So there they all are, but this could go back to 19th century records, as I said. And it does include watch lists, so it's more than what you'll see next is our mere 136. There's been a big disappearance over them, but a lot of those, this map does not map the watch list. There would be an additional 99 if we included the watch list, but still a lot less than 549. So we're losing our orchids, no doubt in part because of nitrogen deposition, which in turn affected manganese. Who'd have thunk it? Not me. Now we get to, we're getting close to the end here, and Alexi's beautiful maps. I need to talk about some more. I just so appreciate Mr. Zanoviev's work here, and I've been sort of encouraging him, you might say, over last couple years to continue on this thread where he's been able to map things at the town level, which I find detail much more worthwhile, delivers much more information to my eyes than the county level maps that we've been able, that is done at the Go Botany site. Not only that, can you see all the distribution? And here again, I'm comparing what you can do with the now lowly map button on CNH, which is all red. These beautiful maps give us this, this difference. Now these are current. The rest are historic past 1980, really historic. And so here's a big change. Now you might think it's a change in sampling, but we're going to see in the next slide, I think, not really probably true. But here's one I would not have guessed, Cirsium uticum, a beautiful fen thistle. You wouldn't think that would be going down the tubes, but apparently it is, at least in terms of the herbarium records. Now this means a couple things. It doesn't just mean that this plant is a danger. It means that you botanists have to get out there and collect these things so we can get more records, so we know what's going on. And there's another scary thing. Right now we're going through species list changes. We're developing the changes to our MESA list. This is not even on there, right? We've got like 12 ideas that we came up with, that we thought, Caro and I, that we thought were really solid ideas. Of course, our bosses think 12 is too many. I should be 10 or maybe 8, maybe even less. We don't know. We're not to the end of the process yet. And then, of course, it has to go through the board review too, not just our bosses. So it make it. But we didn't even consider certain ones that have never been watch listed. But look at that. And again, this is thanks to Alexei Zinoviev, who's been fishing around with this stuff, and somewhat at my encouragement and my suggestions as to editing process here. Now here's 4 of them together, and this comments on one of the things I said before. Sphenophyllus intermedia, not a common plant, not that one that people would normally collect, but see all these red. So these are collected since 1980. So this is sort of evidence in favor of the fact that this is not a result of lack of collections going on in Massachusetts or in New England. These yellows are the result of plants disappearing from the landscape, most likely. Now, you may have, better information on some of these, and over time we're gonna need that. Once again, these are not ones that we selected at all to consider for our even our watch list or our species list. Carex elata, difficult to identify, but it was fairly common at one time, Ovales group. Carex seorsa, really similar in appearance to Carex atlantica. 3 records still on what was more common. Biden's lava, big healthy green lavus on pond edges. No current records at all in the herbarium. And once again, Sphenophyllus with plenty of them in the West. All right, last topic, and we're almost through. And Nancy, you can start the coffee or whatever it is you do. So
No coffee.
I wasn't able to get very far in data, collecting data for this, but it's a cool sciencey thing, and it's fed the nerd inside me. And it's just fascinating kind of stuff. It turns out that across, and you people know this if you've ever done a lot of survey work, if you've been out and taken plots across the landscape, you'll find that at a variety of scales, you will get a lot of repeat customers in your plots. There'll be, low bush blueberry, there'll be high bush blueberry, there'll be white pine, there'll be red oak, over and over and over again. Carex pennsylvanica maybe. And then there'll be, a number of species that are rare. Well, it turns out in plants and in other systems, the vast majority are rare. It doesn't matter if you do 100 plots or 10,000 plots. The vast majority of species by number, in other words, you'll have 20 common species, and you'll have 100 that are rare. They don't occur in many. They might only occur in one plot. Well, that's true in moths too, and many other taxa. It's really bizarre. This really interesting ecologist, Preston, in 1948, did this early work in Orono, Maine, trapping moths at night. And you can see this, he was the first one, I think, or one of the first that used this log scale. This is log base 2. So here you have 2 to the 2nd, 2 to the 3rd, 2 to the 4th, 2 to the 5th, 2 to the 6th, so on. So up here you get your few species, less than 10, that have all these huge numbers of individuals. I assume it was a series of nights of trapping. So the most had like 8,000 come in. And then, down here where you just had 2 during that whole time, you had a lot more. You had 40 species. And you had some species that showed up 4 times. It peaked at 50. 50 species that only showed up 4 times, where you had just a few species that showed up several thousand times. And this is a nice logarithmic curve here. But you wonder, could this curve continue down here? What would that mean? Could you get less than 1 species? Well, you could if you sample more, right? So you would, if you sample another year or 2, you'd push everything to the right. So it would come down and get lower and lower with 1 and 2 and 1 down here, and then you'd sample more, and it would push it out even further. And then maybe that log curve could come down, right? It's very tempting in your statistics to try to get the rest of that curve. Well, that has a name. It's called Preston's Veil. That intrigued me. I thought, that's cool. I got to find out more about that. Here's Preston's Veil with fish in the Arabian Gulf. This crosses all kinds of taxonomic groups. Preston's Veil started here. After 1 month of sampling, they had that same logarithmic curve, base 2. You see it again. They had these 4 species. 1 species occurred 512 times, and so on. But most species were very rare. In this case, 11, only found once. And then they did do what I suggested the last time. They pushed it out to 2 months. Then they got the orange colors there, and they started to curve out the curve, fill in the curve there. They went back across the veil, and then they did a whole year, and they pushed that curve further out and went beneath the veil to find out what was there. So it's a really interesting concept, and I carry it just a little bit further. and reverse the graph. And this intrigued me partly because it turns out my step-cousin in England, who teaches at Leeds, has done a lot of work on this. This is in the Amazon, and he's been involved in all these collecting stations. And they did this really cool paper in Science, and I think it was 2014 or 2013. Terstegi et al. They have a huge list of authors because they all are involved in these data stations. But they were able to find 4,962 tree species. Only trees have to be greater than 20 centimeters in this huge area. But this was a really good data set to test Preston's Vale and to test species abundance distribution, or rank abundance distribution is what they're going to talk about here. And what does it mean? Why is it important? They're going to talk about this here in just a second. But with all these species, they only found 227 that were hyper-dominant. Only, so 1.4% of the total species, but they accounted for the vast majority, more than half of all the trees in the Amazon. How many trees were there? Let's see, does it say there somewhere? I think it says right here. Total number of trees in the Amazon. You've always wanted to impress your friends at parties for this. 390 billion. 3.9 times 10 to the 11th. Isn't that cool? But you can even come close to reasonably assess that number. It is amazing. But they did. They have some great statisticians and use some cool math. Now this flips that same logarithmic graph the other way. So here's the abundance here and the number of, or I'm sorry, here's the abundance here and what's called rank abundance here. So the species that ranks number 1 is the highest in abundance, and in this case it had about 10,000 individuals in all the plots that they took. And then there was rank number 2 was the one just below that, so maybe had 9,500, and so on till you get down. You can see this part of it is logarithmic to base 10 in the number of individuals. So 10 to the first, 10 to the second, 10 to the third, and 10 to the fourth here. And this is a typical logarithmic curve along here. And so the vast majority of species had, less than 10 individuals, and these tiny group had almost all of them here. So how many species should be rare? Maybe, maybe in Massachusetts it should be 500. Well, we sort of have almost 500 if you include our washes, but maybe it should be, closer to 1,000 or something, or maybe it should be less. I don't know. I haven't really done the math, and I haven't got the data yet. Franklin County Flora Group is helping me a little bit with some interesting data, and I'm going to try to get some more too. But the other thing that's super cool about this is this Hubble guy who has this neutral theory, and they calculated out whether their data, empirical data, would match the neutral theory. Now, what's interesting about the neutral theory? What's interesting, and he wrote a big book out about it. He's a professor from Princeton. Big book is in 2001. Neutral theory is a branch of island biogeography. And what's neutral about it is it's in opposition to niche theory. So we often think about this when we're out in the woods or fields or forests or seashore in Massachusetts. Look at how these plants are fitting into these narrow little niches, right? That's why they're rare. This one likes a stream side. This one likes serpentine soil. This one likes a certain kind of sand movement, and that's what they're doing. They're getting into that because they are maybe trying to avoid competition, for example, so they're getting into these really narrow little niches, and that's why plants, well, Hubble said, you can come up with those same numbers without any of that. You can just assume that all organisms, all individual organisms, have the same competitive ability, and then mathematically, all you do is randomly assign presence and absence, and then immigration and extinction, the way they did in island biogeography, and you come up with exactly the same distribution. And there it is. They ran it with all these species, with hundreds of runs of Hubble species. So this is synthetically done with Monte Carlo type simulations then. But amazingly close to the empirical data. So that kind of fascinates me, and turns out Matt Hickler likes neutral theory too. So he and I are gonna tinker around with some of the data and see what we can come up with. And it's what they actually used it for is another form of Preston's veil. Here's their actual data from, let's see, I don't have that, but that same logarithmic graph that they show, but it drops off there. Remember that number, 4,900 some species? That's where it drops off. So if you extend that curve, they can then estimate how many species remain to be identified in the Amazon that are tree species alone, greater than 20 centimeters. And it's huge. Close to 6,000 species are expected to have populations under 1,000. 62% of all species are expected that way. So species richness in the Amazon, they only could find that almost 5,000, but they expect that there's something like 15,000 species there. And they've been doing those plots and survey works for like an entire century. So if you think we have it scary here trying to find stuff, that's a scary in a good way story. Like you could really, so much to discover yet. And I'm going to end with that and this picture of Plantanthera lacera as I end my talk. So thank you very much. It's been extremely yeah, so any questions? Yeah, right here.
The maps with the chemical distribution
Oh, yeah.
of the soil,
Mm-hmm.
is it getting better? Is it getting worse? And
Oh,
why?
it's getting much better. I mean, it's probably mostly because of the fixes of emissions on vehicles, motor vehicles. But it could be some change in farming now. So you can see just in these two, nitrate deposition is much less now. This is 2015. But do you mean like since 2015 or?
I just didn't know if numbers going up or down is good or bad.
Well, I mean, certainly in the 33 years now since the numbers have gone hugely down. They were, well above 20 before. Now they're at 7. This should be our goal, right here, 1. So still more we can do. I'm not sure. I don't know the chemistry that well. I think some nitrate comes from diesel exhaust, and there's probably some other sources from farming or industry. Yeah, but it's a good question. But I mean, in all of those categories, we've made huge progress. There's just still a long way to go. And I think the main effect of plants is like long in the past. But plants and soil have a very long life. I mean, there's still things decaying in the forest from 1985 when all this stuff was coming down. So there's a big legacy effect of this stuff. And I'm convinced that it's had a big effect on orchid populations. And Dennis Wiggum, when he was here, agreed with that, that could be a very likely theory. Yeah, Tim.
I wondered if you had considered amongst some of the threats the possibility of the introduction of the invasive earthworms and the impact they have on invasive species.
So the regular European earthworm I worked a lot with in the upper Midwest, and especially in those northern forests. And we had some surveys where we were required to actually rank the forests. Now this was not the one, there's a newer one that's even more scary that's from China. I can't remember what that's called. Anyone know? The super worm or something? Anyway, the regular nightcrawler that was studied by Darwin, has escaped for centuries because of fishermen bringing it over and then spreading it out and using it as bait. So anyway, we would rank forest stands in the Ottawa National Forest for earthworm effects, and under the theory that certain species of batriciums, for example, would be very sensitive to that, or even trilliums. And I got to the point where I could walk into a stand and tell, like, within the first 20 feet if I was in an earthworm-infested forest or not, without doing any investigation, just by the feel of it under my foot. Now, when we initially did the ranking, you would rank it, and then you would look down and you would dig through the soil to see if you could find evidence of earthworms, or you measure the soil organic matter. But as I said, like towards the end of the summer, I just didn't have to. It was so much softer in the areas without earthworms. Unfortunately, I'd say most, even in the Upper Peninsula, most of the areas were affected in some way. But we got a lot of sort of medium effects, so I don't really know what was going on. There were people in that same area, especially out of Minnesota, that developed this sulfur test. Have you heard about that? Where they would mix a teaspoon of elemental sulfur in a quart of water. Worms don't like that, and they would pour it out on the ground in a fenced area, so that they were measuring a distinct sample unit, and the worms would just come boiling out of the ground with that stuff, and then you could count them, and then you could do sort of various metrics with the soil. And in some of those areas, you could see the advance of the earthworms across the forest soil with the amount of leaves being taken in. I haven't noticed that. Of course, I live in the Worcester Plateau. Compared to the Midwest, I hate to say this, but the soil here in Worcester area is very poor. I mean, I don't know how an earthworm would survive. Certainly not very well. Compared to some of the Berkshire forests, for example, where you do have sweet, rich soil.
That's what I've seen them out there.
Some of the areas too, like along the Connecticut, where, like Coleraine, for example, where a tree falls over and you see this incredible yellow loess in the soil from glacial wind deposits. So there are areas of much soil, but I haven't looked into that much, and it's an interesting question. What do you think you see in Rhode Island?
Some places in Rhode Island have a lot of earthworms, and mostly the richer soil, and I've seen a lot of bad earthworm invasion in the Berkshires in western Massachusetts.
Yeah. So there definitely could be an effect. You're definitely changing the rate of decomposition in the forest floor, and that affects how long the soil's going to hold water, and that will affect, obviously, plants quite a bit. Yeah, I think it's something we should do more of. We should pay more attention to. Probably not in the Worcester Plateau. So, yeah, Brett.
So you said there's some potential changes coming up. You're now looking at things that weren't even listed that, hey, how come these didn't get hit? I understand that regulatory performance standards only allow you to review these species at certain intervals. What would you say we should review this annually, or at what interval should we make this decision?
No, it should be annual. It's too big of a project, and it would not only take too much time and energy, but it would. It's sort of a case of political capital, if you understand what that means. So you can't argue your point too strongly too many times in a row. You have to know how to play your cards and that sort of thing. I think it would be more effective to somehow be able to push for more species every time we do it, which is, I think, a 3 or 4 year interval. Sometimes, it's the first time I've done it. From what I understand, it's 3 or 4 years. Yeah, there's just so much going on in terms of these plant losses, and I think, Alexei, maybe you can speak to this, but I mean, you only really looked for a handful of species and found these examples. You didn't go through hundreds, I'm sure. Yeah.
That's 5 species on the plant list, but we have never seen them ever, so that triggers the search.
Yeah.
Why has 5 species is missing from everywhere around?
Right. That was another thing I noticed in your maps, that the great work that Melissa did in giving S ranks in the 2011 county checklist, and Paul as well. I mean, it's a huge job to S rank everything in the state, and some of these maps that Alexei had were certainly S4s in the county checklist, but based on probably just older data. They certainly were probably S4 at one time. So yeah, Lisa.
I do have a comment about that, though, since I spent a little time in the area, that there are virtually 1% of all the species that we've gotten in the Winterberry for which we have collected more recently than I think.
I should add, when I was studying to do it, I was thinking that the herbarium is not reliable for recent years.
Correct.
So when I collect old CNH part, I was surprised that they changed the picture in many cases. So it was a surprise for me. It shows a picture which may be reliable, not many companies. We have praise.
I think there's a lot more to be learned about that, but I wouldn't be surprised if what you're saying is,
That's
an
why
important point. Pardon me?
That's why we have to collect.
Yes, absolutely, yeah. It's so important, and I'm amazed that some of the more, what we call the more common species, that definitely need to be collected. Yeah, Robert?
Along those lines, and I think what Lisa's saying could be very important too, with the small amount of collecting today. How easy is it for you to put a species on a watch list? Because if Cirsium muticum, for example, was on a watch list, then you'd start to get records and so forth, and you could figure out whether it should be moved on or not.
Yeah.
Is that something you can just do by fiat yourself?
Yes, we're fortunate that we can. We need to do it in sort of discrete chunks because they have to change the list on the website. So we don't want to be doing it every week or something, but.
Every year you could?
Oh,
do
yeah,
a
even
watch
more
list
than that.
change?
Easily several times a year, we could do it.
That'd be good.
And it is important. Some of the things that have been there, though, they lag in people reporting, and we still want to improve our system. Our VPRS system was hard-coded and under contract, so we don't have the code and we can't rewrite it now, but we're working on changes that will make it even easier to report these things, so we can get more information in from watch list species. But yeah, just because it's a watch list doesn't mean it's at all common. It means we need more information. So like the recent split from Plantanthera,
Hyperborea.
one of them that went to
Hyperborea.
Aquilonis and
Hyperborea.
Hyperborea, thank you. Yeah, went to Aquilonis and Huronensis. Once that thing split, you had no information. And it turns out one of them seems to be a little bit more common and we don't need to worry about, but the other one, not so much. Yeah. One more question.
Last
then
question.
we'll-
then
Of
we
the
should-
hundreds of reports you got this year, how many come from state employees or state contract versus volunteers?
The vast majority would be from volunteers, but we do probably a couple hundred. I think we did
You and I did about 150 this past
Yeah.
year.
yeah.
And of the volunteers, how many go through NEPCOP first?
Well, there's NEPCOP and there's PCVs. There's a lot of reports that don't come from them. We get Franklin County Flora reports. We get Peter Grimos, who's not here, forester out in the Berkshires, lives in Greenfield, amazing botanist. We get a lot of reports from him. People like Charlie Quinlan, who are consultants. Amanda, when she was a consultant here, maybe Matt too. Lots of reports from
Brett.
Brett, sorry.
Except for Brett hasn't
Looking
sent
at
very
the
many
collection.
plant records lately.
No, Brett has done some terrific records that are amazing. So, yeah, sorry about that.
That's okay. We do have to wrap up. Sorry we started
Yeah.
so late. More questions for Bob. We'll have some refreshments next door, but thank you all for coming. We didn't want to stop it in.